Hi, I'm James Rodeer, and welcome to the American Institute of Biological Sciences Bioscience Talks, a forum for integrating the life sciences, where we discuss the latest bioscience publications. As a reminder, if you'd like to read more, point your browser to bioscience.oxfordjournals.org. And for our third episode, I'm joined by two researchers from Harvard Medical School, Xandra Breakefield and Mikolaj Zaborowski. They study extracellular vesicles, a field that has profound implications for intracellular communication, disease diagnosis and treatment, and a myriad of other topics, as we'll soon hear. We had quite a bit to talk about, so let's get straight to the interview. Dr. Breakefield, Dr. Zaborowski, thank you very much for being here today.
Yeah, we're happy to join in on the call and talk a little bit about the vesicles and this new form of intracellular communication.
Great. And just to get us started, for listeners who may not be familiar with extracellular vesicles, just what are they? What are we talking about here?
So extracellular vesicles are vesicles that are released from basically all cell types. They are very variable in size, and we distinguish many different types of vesicles, but in general, it's a structure that is a part of the cell that is membrane bound, and it may contain different contents of the cell, including proteins, DNA, RNA, and all lipids that are bioactive in the cell too.
Okay, so we're talking about vesicles that are many times smaller than a cell in most cases, and they're carrying lipids, protein, genetic information. I guess the next logical question is, why are the cells releasing these?
Well, it's a really relevant question, and there are many people trying to answer it, and so far it has been shown that these contents can be biologically relevant. It means that RNA that is released from one cell can get to the other cell, and it is possible that it regulates the function in the recipient cell. And it's similar also for proteins, lipids, and potentially DNA.
So this sounds a lot like an intracellular communication system where the cells are releasing an entity, and then that entity is being taken up by other cells where it's having a biological effect.
Yeah, I mean, basically, it's like, the Earth sending off a satellite onto, the moon, and it's going to pick up signals from that, but it's also going to change the moon's environment somehow. And the things it can carry are very different than the type of things that a cell could release. Like a cell can release certain proteins, but it can't really release free nucleic acid. It would just get degraded immediately. So by putting it in this vesicle, you protect these highly charged molecules that aren't able to pass just through fluid. That they need to be protected in the fluid space, and then you have the kind of the power of the genetic code and all the genetic manipulations that are possible in a cell can be transferred from one cell to another.
So the cells are releasing this material through the extracellular vesicles, and it's having a biological effect in the other cells.
Yes. That's correct. And there are many examples of this type of biological effects. Mainly, they were studied in the communication between cancer cells and also between cancer cells and host cells. But there are also many other biologically relevant events, like they have been studied also in inflammation or in neurodegenerative processes. So they are involved in many different types of biological events.
Yeah, I mean, I would say just in terms of neurodegeneration, if you think of something like Alzheimer's disease, there's another function that these vesicles, or some types of these vesicles can have, which is to try to get rid of the garbage of the cell. So if you have, a cell that's accumulating A-beta and it's toxic, the cell will try to get rid of it in vesicles, but then what happens is those vesicles, within the brain just fuse with another or are taken up by another neuronal cell and just accelerate the degenerative process.
It occurs to me that these are appearing in an awful lot of cell types. Is it most cells or all cells that release vesicles?
It's actually very hard to find the cell that does not produce vesicles. So usually we are able to isolate them in various quantities from different types of cells.
It's also expanded to include the whole universe of life forms. So bacteria that we're now known to secrete vesicles, and viruses they do it through the cell, will secrete vesicles. But there seems to be this type of communication is going on even in the lowest unicellular organisms.
And speaking of this type of communication, when was this first discovered? This sounds like it has extremely wide-ranging implications, and I'm wondering, why didn't we know about it until recently?
Well, I think there were kind of two issues. People could see, for instance, in cultured cells, this kind of floating stuff, but the standard microscopy in labs wasn't really high enough resolution, so they called it cell debris. And there was a big debate about whether it was just debris or it was vesicles.
And then who were the first to demonstrate that there really was something more to vesicles than simply cellular debris?
Well, I think the best example I know of that is the group in France. It's Clotilde Thierry and Gracia Raposo. So they study immune responses, and they grow dendritic cells in culture. And they notice the vesicles, and they characterize their protein profiles, and they characterize them by electron microscopy. And they showed in different ways how different proteins, like they have MHC class 1 and class 2 antigens on their surface, how they could not only present antigens, but affect the function of immune cells, other immune cells that encountered the vesicles. And I think the immunologists were very interested in them at that point, but it was only when the RNA was discovered in them, which was initially done by Jan Lottwall in Sweden, again using normal cells, that people said, "Oh my gosh, you could actually transfer genetic information through these vesicles." And then our group was the first actually to show that they could be used as biomarkers for a disease state.
And speaking of disease states, cancer seems to come up rather frequently in discussions of vesicles. I was hoping you could talk a little bit about oncosomes and their role.
Oncosomes are one of the vesicles that have been shown to be released by cancer cells. Actually, all the previously mentioned vesicle types are also released by cancer cells. So altogether, vesicles may enhance the proliferation of cancer cells. I mean, vesicles released from one cancer cell can get to the other one, and then this way it may cause increase in proliferation. They may also change the phenotype of recipient cancer cells in a way that will promote invasiveness of these cancer cells.
I think the oncosomes do seem quite distinctive to cancer cells, that they're very large, but in fact, we don't really know which vesicles the cancer cells are releasing or causing the real problems. But the other things they do are they stimulate angiogenesis. They have enzymes on them that eat into the extracellular matrix to help. They have kind of different methods of depressing the immune response to the tumor. So they're a very critical part of a cancer's success. I think if you took the vesicles away, it would be, but it's hard to do that, would really compromise the ability of the cancer to take over.
That's fascinating. It raises the obvious question. If the vesicles are being used by the cancer to proliferate, is there any research being done into slowing or stopping that mechanism?
Well, that's a very interesting question and very active field. The main challenge here is that since vesicles are so important to the function of host cells, it's very challenging to stop the production of vesicles only in cancer cells.
So any therapies along those lines would be something we would look for in the more distant future?
Well, I've heard there's a company in California that tries to do like plasmapheresis and just take the vesicles out of the bloodstream to see if that will slow the cancer down. I don't know how that's going. There's another whole arm of this where people say, gee, these vesicles are so great at delivering things. Maybe we can use them to deliver drugs or, microRNAs that would stop tumor growth. And so there's a lot of work or different proteins that are therapeutic. So there's a lot of work going on trying to load vesicles and have them be, delivery vehicles for anti-cancer agents.
And how would that work? Would the patient take a drug of some sort, or would the cells be manufactured outside of the body?
I think that most effort is going into creating, clinical grade factories of normal cells. Mesenchymal stem cells are very popular. They come from the bone marrow. Loading them through genetic engineering, loading the vesicles they produce, and then harvesting those vesicles and then injecting the vesicles into people.
And are there therapeutic avenues other than in cancer that are currently being explored?
Oh, yes, very, very actively for cardiac damage. It seems to be very effective. We're kind of cancer people, so we don't. But we come across there's plenty of other things, other diseases that are being explored. Arthritis is one of the areas that looks very promising. Kind of suppressing inflammation in certain areas.
That sounds like an amazingly wide range of possible applications. One thing I did note in your article, though, is you mentioned that these vesicles are hard to work with. I was hoping you could explain some of those difficulties.
So one of the biggest challenges in the field is actually the method of separation of different vesicle types. So though we try to study different vesicle types, it's really by enrichment of one of them, usually, because so far among different isolation techniques, there are three that are usually used. It's ultracentrifugation. This can be also sucrose or iodixonal gradient, and the third one. These can be also immunoaffinity methods. There is, I should say also, there is a fourth one that is that becomes more and more common. It's size exclusion chromatography. So these all methods they allow you to enrich one of the vesicle types, but not really get rid of all others. So that's one of the challenges. And the other one is also that it's also related to methods of isolation. Sometimes it can be difficult to distinguish the effects that are caused by vesicles itself and from the effects that could be caused by proteins or any other active molecules that are in the conditioned medium or in biofluids.
And the other problem is that there's so much information in the vesicles that it's very hard for you to figure out what component actually caused the effect that you're seeing.
Okay, so we've talked some about the difficulties in working with vesicles, and earlier we spoke about the therapeutic potential, but do they also have potential as a means of diagnosing disease?
Oh yes, definitely. That's very
That's
active.
A major
question.
major effort. Yeah.
Yeah,
in the field.
Yeah, that's something we also focus on in our lab. Is how to use the information from RNA or DNA to try to characterize some signature that would be characteristic for cancer cells. And, well, the ultimate goal of it is to be able to characterize this signature in biofluids from the patients. So the ideal future achievement would be to be able to isolate vesicles and then, for example, mutation or some RNA that is characteristic for cancer cells from human plasma or any other biofluid.
Yeah, it kind of merges with the precision medicine initiative, and especially for cancer. Each person, they can have the same type of cancer, but really they can have different mutations that are driving it. And the physician needs to know which are the critical drivers of that tumor. And they're going to change over time. So especially in the case that we work with brain tumors, the physician can't keep going back in and biopsying the tumor as he's progressing through his treatment and trying to figure out what's happening. So they want to read out, that they can do longitudinally in a patient under treatment to help them decide, is the tumor, growing? This would also be true of a metastatic tumor where you don't even know where all the foci are. Is it growing? Is it changing its genotype now that I treated it with this one agent? Is it now turning into something else? And is there another drug I can use to treat it? this is very close to becoming products for some forms of cancer. So, I mean, there's a company that actually sprung out of our lab that we're not directly affiliated with it, but called Diagnostics, that has a couple of products for cancer detection, for lung cell cancer, small lung cell cancer, and I think prostate cancer that are very close to becoming actual products. And the NIH is very vigorously pursuing this and trying to set up big reference databases because you need to compare it to, and it's being done not just for cancer, it's being done for practically any disease you can think, high risk pregnancy, neurodegenerative diseases, heart failure. I mean, graft versus host, everything, because it's with a simple, typically it's taken from the blood, but you can use other fluids. You can use saliva, you can use urine. They all have vesicles, and a subset of the vesicles will be derived from the diseased tissue, and then it helps you to monitor the state of the disease.
Okay, so we're looking at vesicles that are derived from more easily accessed bodily fluids. Does something like this have the potential to replace a biopsy?
Oh, yes, definitely. That's the goal.
Wow, that alone sounds like a really big deal. Before we closed, I was hoping, because this is such a new area of study, that I could ask you a couple of speculative questions. And one in particular we were wondering about, is there any role of vesicles that's been seen or studied in reproduction?
Well, there's a very interesting study. I forget the name of the fellow. He's in Britain, but the actual process of fertilization is a vesicle-mediated phenomena, where the eggs produces vesicles at the point of sperm contact, and that changes the surface and allows the sperm in, and then also is involved in preventing other sperm from entering. There's a whole story on that.
All right. Next up, neural cells. We know that they release a lot of vesicles. Is there any thought at all that they may have an influence somehow on behavior?
Well, I mean, it changes. It has a role apparently in synaptic plasticity and formation of synapses. So to that extent, it's involved in reinforcing a certain pathways, but I don't think it's in any way selective for any specific type of behavior, motor behavior or emotional behavior. It's happening in all the neurons.
Right.
Now, but we have thought about, this is really, and I don't know if we should even, for instance, we think that, some people think that addiction is caused by changes in genome methylation patterns. And that's why withdrawal takes so long, because you have to actually change acetylation and methylation patterns of the DNA, which determines its gene expression. And that potentially, that's one of the ways vesicles could be used therapeutically, is to bring in molecules that would speed that process.
So there's work ongoing, but it's still speculative.
Right.
And vesicles are released by plant cells as well. Is there any work being done in this regard, be that agricultural or otherwise?
You know what? There's a guy in Kentucky whose name I forget, who actually, this is very interesting. So because, there's so many limitations that we undergo in getting clinical grade products that come from mammalian cells, he actually makes vesicles from different types of fruit. He makes vesicles from grapefruit, grapes, and he loads them with drugs and stuff like that. And those can actually be given to patients without going through all, because they're natural products, right? So he does have trials where he gives them to patients. From talking to him, my impression is that like for people with oral cancer and they're on chemotherapy, you can't take away their normal therapeutic, regimen, but I guess their mouths get very, very dry, and he actually finds that giving them these plant vesicles really makes them feel a lot better, and they get normal salivation back and stuff like that.
That's great. And I don't know if you'll be able to answer this, but if you are, what's next for your research? What are you working on right now?
Well, I don't know if I can tell you that. I don't think Mikolaj can't tell you what he's doing, but we are trying to, so in general terms, we're trying to increase the sensitivity of these biomarkers in blood for cancer, because we want early detection And right now, that's something that's hard to achieve, and that's very critical. Let's say, for example, in ovarian cancer. The problem for some of these cancers is that you don't detect them until the person is very well advanced. That's also true for glioblastomas. So we're trying a lot of exotic ways, taking advantage of the RNA that's in these vesicles to try to increase that sensitivity. We're also we and others are trying to selectively capture out the vesicles from the tumor cells, so we get rid of this normal RNA background. So biomarkers is very big. We're looking at a lot into how especially the brain tumor cells change the cells in their environment in support of tumor growth through the transfer of these vesicles. And we have some data already on that. We continue to work on that. And we're also working in the therapeutic arena trying to load them, figuring out how to load them with specific RNAs, how to load them with specific proteins to use them for therapeutic applications, primarily in our case for cancer.
So potentially one day we may use vesicles to give a blood sample to find out if we are sick, and then if we are sick, we may have other vesicles that deliver our care.
That's right.
That's a very exciting future. Dr. Breakefield, Dr. Zaborowski, thank you very much for being here.
Hey, thank you. It was great talking to you.
Thank you so much.
And that concludes this episode of Bioscience Talks. Just a reminder, the journal Bioscience is published by Oxford University Press on behalf of the American Institute of Biological Sciences. To read the article we talked about today, point your browser to bioscience.oxfordjournals.org. Thank you for listening. Talk to you next time.